Welcome everyone, this is a Legacy School Science lesson for Biology Chapter 6 about classification and variation. This is part 1 of 2 parts. For this part you will need your Science Checkpoint Book 3, pages 86 to 96. When you come across an unfamiliar animal in a habitat survey, you might look at it to see if it reminds you of anything else. If it has 6 legs, you might compare it with an insect that you know. If it's a bird, you might compare its plumage with a bird you are familiar with. This comparing, looking for similarities and differences, led scientists to develop keys and to classify all living things on Earth into groups. The main groups of living things are microorganisms, the plant kingdom, and the animal kingdom. The animal kingdom is then divided into 2, vertebrates and invertebrates. Microorganisms. Microorganisms can be divided into 4 groups. These include fungi kingdom, Monera kingdom, Protozoa kingdom, and viruses. Table 6.1 shows the features of each group alongside example of each. The plant kingdom. The plant kingdom is made up of different groups, some of which are algae, liverworts and mosses, ferns, conifers, and flowering plants. Table 6.2 showing features of these groups alongside examples of each. The animal kingdom is divided into 2 groups, the invertebrates, which are animals without a backbone, and the vertebrates, which are animals with a backbone. The invertebrates. The invertebrates have many types. Some of them are the jellyfish, the flatworms, the annelid worms, the nematode worms, the arthropods, and the molluscs and echinoderms. Table 6.3 shows features of each group and example of each one. The vertebrates. The vertebrates are divided into 5 groups. These are the fish, the amphibians, the reptiles, the birds, and mammals. Table 6.4 shows vertebrates. There are 5 groups of vertebrates: fish, amphibians, reptiles, birds, and mammals. Table 6.4 shows the features of each group alongside an example of each. Keys. There are 2 kinds of keys: spider keys, whose name refers to the way the keys spread out like the legs of a spider, and numbered keys, which take you step by step to the identification of the specimen you have found. Keys are made by considering features in a species that do not vary. Features that might change due to growth, for example, are not used. Spider key. On each leg of the spider is a feature that is possessed by the living thing named below it. An example is shown in figure 6.5. A spider key is read by starting at the top in the center and reading the features down the leg until the specimen is identified. This is an example of a spider key. As you can see, it starts off with the broad-leaved trees. And then the broad-leaved trees is divided into 2 parts, which are the single leaves and the compound leaves made of leaflets. In the single leaf, we have the wavy edged and we have the unwavy edged. If it's wavy edged, then it's an oak tree, and if it's unwavy edged, then it's the beech tree. For the compound leaves made of leaflets, we have leaflets attached to the center and leaflets arranged in pairs. If it's attached to the center, this is a horse chestnut, and if it's attached in pair, this is ash. Numbered keys. You work through a numbered key by reading each pair of statement and matching the description of one of them to the features you see on the specimen you are trying to identify. At the end of each statement, there is an instruction to move to another pair of statement or to the name of a living thing. Here's a simple numbered key. It can be used to identify molluscs that live in freshwater habitats such as river, lakes, and ponds. So in our example, you start by first clue. Is it a single shell or is it a 2 shell? If the specimen you have is a single shell, then you will see clue number 2. If the specimen you have is a 2 shell, then you will see clue number 6. Let's say that the specimen we have is a single shell. Then we will move on to clue number 2. Is it a snail with a plate that closes the shell mouth? Or is it a snail without a plate that closes the shell mouth? If it's a snail with a plate, then this is Benethia. If it's a snail without a plate, then we will have to move on to clue number 3. So if it was the snail with the plate, then we have identified our living organism. But if it's not, then we will have to move on to clue number 3. And then in clue number 3, we'll see that it says a snail with a twisted shell, a snail without a twisted shell. So if it's without a twisted shell, then the organism is a freshwater limpet. If it's with a twisted shell, then we have to see clue number 4. And so on and so forth. You keep going down the clues until you find the living organism that you're looking for. Inherited characteristics. The features of living things that we have looked at when considering adaptation and making keys are also known as their characteristics. When you look at different generations of the same species, you see that they have a large number of the same characteristics. It appears that the characteristics are passed on from one generation to the next. The latest generations has inherited its characteristics from the generation before. Once scientists grasped this idea, investigations into inherited characteristics began. George Mendel and Hugo de Vries. George Mendel was an Austrian monk who studied mathematics and natural history. He set up experiments to investigate how features in one generation of pea plants were passed on to the next. Pea flowers self-pollinated. When Mendel wished to control the way the flower pollinated, he cut off the anthers of the flower, collected pollens from another flower, and brushed it onto the stigma of the first. He completed his task by tying a muslin bag around the first flower to prevent any other pollen from reaching it. Mendel performed thousands of experiments and used his mathematical knowledge to set out his results and to look for patterns in the way that the plant features were inherited. He suggested that each feature was controlled by an inherited factor. He also suggested that each factor had 2 sets of instructions and that parents pass on 1 set of instructions each to their offspring. Many years later, it was discovered that Mendel's factors were genes. Mendel's work was published by a natural history society, but its importance was not realized until 16 years after his death. At that time, Hugo de Vries, a Dutch botanist, had been studying how plants pass on their characteristics from generation to generation. He was checking through published reports of experiments when he discovered Mendel's work. He found that his work supported Mendel's, and after studying Charles Darwin's hypothesis called Bangenesis to explain inheritance, he went on to suggest that information about the characteristics to be inherited were in the form of small particles called Bangenes. Darwin's Bangenesis hypothesis was shown to be incorrect due to Vries' idea about Bangenes continued to be useful to scientists studying inheritance, and a Danish biologist called William Johansen made the word shorter and called the inherited particles genes. Cells and reproduction. As living things are made from cells, scientists began looking at cells for evidence that they may be involved in reproduction. We have seen from slides of tissue that cells have a cell membrane, cytoplasm, and a nucleus. When scientists examined living cells under the microscope, they saw that changes took place in the nucleus that are linked to reproduction. Just before a cell divides, a long strands of material appears in the nucleus. These strands are called chromosomes. So these are the chromosomes that divide, and then when we have the 2 gametes, they separate. So we have the cell, as we know, it has a cell membrane, a cell nucleus, and it has chromosomes and inside the nucleus. And then we have the nuclear membrane. In the time before cell division began, when chromosomes are not visible under the microscope, each one has made a copy of itself. And the chromosome and its copy lie together as two threads. During cell division, as the nucleus divide, each pair of threads separate. Each thread then enters one of the two nuclei that are forming in the new cell. Once each cell is formed, every thread becomes a chromosome. This means that the nuclei of the new cell have the same number of chromosomes as the original one. The chromosomes in the nuclei of the new cell then seem to disappear into the nuclear material but become visible under the microscope once more when the cells are about to divide. Chromosomes and genes. Extensive studies on chromosomes have shown that they are arranged in pairs in the nucleus, and individuals in each species have a certain number of chromosomes in their cell nuclei. For example, humans have 46 chromosomes arranged in 23 pairs in every nucleus, and a fruit fly has 8 chromosomes arranged in 4 pairs. The chromosomes are really threads of chemical messages. These messages are strung along each chromosome, like the carriages in a very long train. Each message is called a gene. The gene provides all the information for how the cell grows, develops, and behaves, and how the body grows and develops too. Each pair of chromosomes has pairs of genes that carry information for a particular characteristic, such as eye color or hair color. These genes are situated at the same point on each chromosome, as figure 6.11 shows. So as we can see, we have the two genes on a pair of a chromosome, and the gene for the eye color, they exist on the same location in both genes. This is the end of the first chapter. I'll see you again soon.